Hello students, I hope you are getting the idea of the whole process of drug discovery and development. In this part of the drug discovery and development, this is drug design, we will be seeing the pharmacokinetic parameters of lead compounds and how we can optimize them to make our desirable clinical candidate. So here is an example, like if our target is a receptor which is a FGFR fibroblast growth factor receptor, which mainly drives cancer in bladder cancer. So if our target is this receptor FGFR, so we have to develop a small molecule drug. So in this example, this is the lead compound which was developed against this target FGFR, and after some modification at the drug design phase or after some modification in the pharmacokinetic properties, so this in the right side, this is the modified drug or clinical candidate, because it was not entered in the clinical trial, that means human trial, but it is referred as clinical candidate because it showed potential favorable profile in the preclinical tests. So this is the lead compound, and you can see this, it has some profile, like these are the pharmacokinetic properties or parameters, like this is IC50. You know what is IC50? This is a measure for potency. How potency is it? Two IC50 values. One is this is at 0.7 nanomole concentration, this lead compound is inhibiting the FGFR receptor, and this PFGFR is the phosphorylated version of the receptor. So the receptor maybe undergo phosphorylation or dephosphorylation. So it is necessary if our target undergoes phosphorylation or not. So in the body or in the activated state, the receptor might be in the phosphorylated state. So the phosphorylated FGFR inhibition concentration of the IC50 is 19 nanomole. So these are the potency measures of our lead compound, and this is the molecular weight 449.5. Log D is the lipophilicity 3.9. Solubility, water solubility is 27 micromole. Molar solubility, you can say. Clearance, intrinsic, so this is intrinsic clearance in rat. So it is in vivo, in the mice model or in the rat, not in the human. So clearance is 60 microliter per minute, and this F, percentage F, is the bioavailability, which is 50%. That means if we ingest 500 milligram of a drug in the oral route, that means only half of that, 250 milligram of that drug will be available in the blood. So another important parameter is this one, HERG. So we will discuss more about this HERG. It is another receptor which is mainly harmful. So if our drug mainly inhibits this receptor, which is mainly associated with fatal cardiotoxicity, which is not good. So our drug could inhibit this receptor, but it needs to be at high concentration. So 30 micro is, you're saying that 30 micromole of our drug or lead is required to inhibit the HERG receptor, whereas the therapeutic dose or therapeutic potency, you can say, is 0.9 nanomole, so which is quite safe. Here is a lot of gap between this 0.7 nanomole and 30 micromole. So we will be seeing this more in details, but the higher the value of HERG, that is more safe. So this profile, and also in the right side, this profile is quite similar, but the numbers are critical here. You can see the profiles are very similar, but the numbers are critical. What does that mean? So that means after some modification we changed few structural changes. So you can see mainly we reduce the lipophilicity here. So if we reduce the lipophilicity, a lot of things will change, like it will increase the clearance, or it will increase the permeability, it will increase the um, bioavailability. So in this specific example, the scientists, they mainly reduce the lipophilicity here. So they reduce the lipophilicity because there was some problem in the solubility. So this, you can see this solubility was 27 micromole. So this is not quite high. So if you can see this, if we take in the oral route, the drugs needs to be undergo dissolution first. So for it to undergo dissolution, it needs to be highly soluble or moderately soluble in the water. So at some level of solubility is required. So if you see this here in the right side, the solubility is high. It is quite high. From 27, it is 157. So maybe this 157 is enough for water solubility in the stomach or in the GI tract. But you can see this, as a result of high solubility, the log D, the lipophilicity has decreased. You can see this 3.9, 3.1 to 2.4. So the lipophilicity is decreased, and we know that with lipophilicity, if lipophilicity is decreased, it will also decrease the potency. It will also decrease potency, and it also reduce the metabolism. So that means reduce the clearance rate. So you can see the clearance rate here was 60 microliter, but it reduced to 39. So this is quite another important fact, because this feature, like this is 60 microliter, that means it is quite high, highly high. That means the liver is clearing the blood like 60 microliter per minute. So if a drug remains like not insufficient in the blood, that means it is quickly removed or cleared, that is sufficient for a drug to fail in the clinical trial. So we need a at least moderate amount of clearance rate, or the clearance rate should not be higher. So here in the right side, you can see this clearance, intrinsic clearance is decreased. So it is quite safe, because as clearance decreases, that means we need less amount of the drug, less dose, so which is also safe. So high dose is not desirable here. Also note that the downside is this bioavailability, F has decreased. So this is one downside, because from 50% it has reduced to 42%. So we have to maintain a balance here. So one property is like F is decreased, but other few properties has improved a lot drastically. For example, the solubility has improved manifold, the clearance rate has decreased a lot. So these are desirable. Also note that 3 unit of the Hertz, this HERG IC50 value has increased to 3 unit, which is also good, safe. So we will discuss later, but you get the idea, like what are desirable and what are undesirable properties of the drug in this optimizing PK optimizing phase of the drug design. So moving to the next slide. As you know that the goal of our drug development is to develop a safe and effective drug. So those two are the main goals. Safe, it has to be safe, and it also has to be effective. So those two are the main criteria. So mainly we test our drugs in different screening methods, and we get we get data, and then we analyze the data, and after analyzing the data we come to the conclusion that whether this drug needs to be taken in the next phase, which is the clinical trial. So based on those data in the preclinical trial, we mainly take the decision that the drug would be safe or not, or effective would be not. So you can see this, a lot of the cost, like a substantial huge amount of the cost has been like cost has been expense is on the clinical phase. You can see this phase 1, phase 2, phase 3. Phase 3 has the largest amount of cost involved in a drug discovery development phase, and then phase 2 is moderate, and phase 1 is also moderate. But preclinical, you can see this is the preclinical cost, which is very less or negligible comparing to the whole phase 1, phase 2, phase 3. So we need to be very careful here in the preclinical trials. So our data needs to be sufficiently good to make us the decision to move forward or not, to go to the clinical phase or not. So in this phase, throughout the whole phase of the preclinical trials, we get results from different sorts of tests, and by those results means data, by analyzing those data, we mainly take the final decision to go in the clinical phase or not. So ADMET mainly refers to the pharmacokinetic properties, absorption, distribution, metabolism, excretion, and toxicity or toxicology. So these are the basic stuffs, not that complicated. Absorption means the drugs needs to be from site of administration, that means the oral route. The drug needs to go in the plasma from the GI tract if that's oral route. So it has to cross a lot of barriers. A lot of factors depend on those transfer of the drugs or absorption of the drugs to the plasma, and then after reaching the drug to the plasma, the drug distributes throughout the whole body. That means tissue distributions and other distributions, like in the blood or in the CSF or other liquid, different compartments of the body. And then metabolism is like it's occurring concomitantly when drug is distributed and metabolism occurs. So, and last, the excretion. After metabolism, the drug excreted throughout the kidneys mainly. So toxicology, it is like the metabolites are toxic or not. We know the drug, we are testing the drugs, but it is not always possible for account for its all the active metabolites. So we also see the active metabolites which are toxic or not after the drug is metabolized. We also test like general toxicity, like that is the drug is cardiotoxic, or it is like carcinogenic or not, or it is causing any damage to the DNA or not, or is it causing inhibiting the cardiac channels, potassium channels or not, the HERG channels or not. Those are fatal toxicity. So those also needs to be addressed first before the PK parameters. Actually, the whole process goes hand in hand together, like the in vivo models. We see both of these things together, like toxicity and ADMET properties. So going to the next slide.